my name is Ricardo Harapal, and I'm here on behalf of the René Salazar Speaker Series. So here at the Broad, we have a Latinx community, and it's called Latinx at the Broad. And we have the mission of building community among Latinx Broadies and allies and empowering members of the Latinx communities. We also do social events and professional development events for our community. The René Salazar Speaker Series is one of the programs that we run, and we want to highlight Latinx and diverse researchers within and outside of the Broad. We want to foster collaborations and allow visibility and role models for diverse individuals, and we want to allow opportunities to network and promote research here, abroad, and internationally. So here now I want to welcome our speaker, Dr. Juan Alvarez-Dominguez. And just as a brief bio, I wanted to mention, so Dr. Alvarez is an professor of cell and developmental biology at the University of Pennsylvania Pullman School of Medicine, and he's a member of Penn Institutes for Diabetes, Obesity, and Metabolism, Chronobiology and Sleep, and Regenerative Medicine. Dr. Alvarez's research focuses on the interplay between metabolic cycles, circadian oscillators, and islet maturation. This work involves single-cell and high-throughput experimental and computational approaches with a view to rationally engineer mature stem cell-derived islet organoids as human models of islet development and physiology, and as a novel replacement therapy for insulin-dependent diabetics. Dr. Alvarez completed a Howard Hughes Medical Institute LSRF Research Fellowship with the Melton Group at Harvard University, asking how circadian rhythms shape islet development in diabetes, and using human organoids and mouse models to study these effects. He received his PhD in biology from MIT, where he studied the modulation of lineage-specific cell differentiation by long-coding RNAs, and his AB in molecular biology from Princeton University, where he studied the role of DNA self-catalytic depurination in site-specific mutagenesis. So take it away, Dr. Alvarez.
Thank you, Ricardo, for that kind of introduction. I will share my screen. All right. So it's a pleasure to talk in this series, and I was told to do two phases. So this is the roadmap for my talk today. I will cover first a little bit about my journey, and then I will move on to the research that we do. So shall we get started? The first component is going to involve maps. This is where I was born. I was born in Guatemala, and I was born near the coast. So here's a more detailed map. We're close to Mexico in the border, and we border with the Pacific Ocean. And the first trip that I wanted to highlight is a trip that I took on weekends to our capital city, where I was studying mathematics classes. And so we will board the chicken bus, which I think are high school buses from here in the US that have been refurbished and repainted. There's a nice documentary about it on Amazon. And then, this was a sacrifice because it involved traveling several hours on my own, but there was a reward for it. I became awarded in biology and other disciplines. And the first prize that I got for it was a PlayStation. That's what I did with the money I received. But there was something else that this sacrifice actually rewarded me with, which was the opportunity to go study abroad. And so when I was 16 or around 16, I got my passport and I went to the United World Colleges. There are several of these colleges around the world, but the one that I went to was located in Italy. These ones are sponsored by the United Nations, and they take on full scholarship. All of the countries in the world nominate one student, and then there's one student from each country. So I was the one student from Guatemala. These are usually castles by the sea, and they are, as I mentioned, all the students are on full scholarship. So this was my high school where I would do my swimming. We didn't have a swimming pool, but we would swim in the ocean. And then this enabled me to apply for schools abroad that also studied in English. And so I came to the United States in 2005 for undergrad, and that was the first time I've been here since. So in the United States, I was able to also take advantage of scholarships, and therefore I was able to go to MIT for grad school, as Ricardo was mentioning. I also went to Princeton, where I know Carmen went, and then I did my postdoc in Harvard University. So I often get the question of why I only went to Ivy League plus schools, and the answer is very simple and clear. Only they had the money to support full ride for somebody who couldn't afford it from an international precedence, because I was not a resident or a citizen in any of these institutions. So then the last part of my journey has been coming to the Perelman School of Medicine at the University of Pennsylvania, where I have my group of students. My lab is located at the Perelman Institute of Translational Research, where we have several people working in multiple disciplines, but we also see patients. And then here's an early photo of lab members. This is a good photo because we have an undergrad, master's student, and a graduate student here who are celebrating the undergrad finishing classes. And so we're located in Philadelphia, and I'll be happy to answer any questions about my journey at the end of the talk. But before we do that, I want to move onto the second part of my talk today, which will be about the research. So I'm very excited to share a little bit about the research. You're all scientists, and you all know that I'm going to talk today about circadian rhythms. And so I want to give you a refresher of what I mean by that. In order to make a general enough statement, I will say that this is something that is experienced by all organisms on Earth, and it has been the most consistent stimulus that we have experienced in our evolution, and that is simply the rotation of the planet. Because the planet rotates, there are cycles in, for example, in light and dark that are unavoidable, and those cycles have consequences. For example, our harvesting energy during the light cycle, if we're diurnal, or during the night cycle, if we're nocturnal. And this also has consequences for temperature and other parameters of the environment. And so most organisms on Earth have had to deal with this and have adapted their physiology to the rotation of the planet. If we would have been born in Mars, then it wouldn't be 24 hours. It would be according to the time it takes for Mars to rotate around its axis. One cool consequence is that your pancreas is an organ who has to buffer these fluctuations in order to keep your blood glucose constant. And it does so across periods of feeding and fasting. So at night, you are not eating because you're sleeping, and that's considered fasting. And during the day, you consume meals. So the pancreas buffers these changes with its alpha cells, which make the hormone glucagon, which, if you remember, it raises blood glucose by making the liver produce it. And the counterpart is the beta cells that produce the insulin hormone in order to lower your blood glucose after a meal. And so I wanted to highlight the two functions of the endocrine compartment of the pancreas and how they need to buffer fluctuations across feeding and fasting cycles, because that will become important later in my talk. And so this basically, this organ acts as a rheostat, which is the machine that keeps the temperature in your house. And the context in which we'd like to study this rheostat is specifically in the context of beta cells. The beta cells are the ones who I told you produce insulin, especially in response to increases in glucose, but they have degrees of maturity. When you are just born, you're neonatal, your beta cells are immature, and they respond to small amounts of glucose with small amounts of insulin. However, later they develop the capacity of kicking out much more hormones. So their hormone release capacity increases, and they are considered more mature because they also reduce their basal activity and only respond to stimulatory concentrations of glucose. And this becomes important for diabetes and for making diabetes replacement therapies. However, this process can also be reverted, so it's a plastic process. And in the context of genetic lesions or circadian perturbations, or also through the progress of diabetes, this mature function regresses into a more immature state. And we are interested into knowing how the acquisition of circadian rhythms is important for this balance between immature and mature functions. And so I'd like to label that as chrono-energetics. And what my lab is interested in is studying the link between the maturation process of beta cells and how this harmony with the external energetics of the environment is negotiated. So in order to give you a little bit more background, I have to mention that there's a timekeeping mechanism in all of our cells. In fact, it evolved since bacteria. And so this timekeeping mechanism is called the clock. It consists of activators and repressors that establish a feed-forward loop with a 24-hour delay. And this is how your cells are able to keep track of 24-hour periods independent of the external environment. This is a cell autonomous function. And so people call this a clock. And what this results in is that it creates oscillations in the expression of most molecules in the cell, including mRNA and protein. And it receives input. These are considered the outputs of the clock system. And one of the things that the circadian clock does is align these oscillations so that they're peaking at the right time of day. For example, cell replication peaks when resources are abundant in the environment, and DNA repair peaks when the roadblocks for making new synthesis of nucleic acids are abundant. Other inputs that align these oscillations include light, feeding, exercise, aging, temperature. And what these do is that they align the phase. They do not drive the oscillations. As I mentioned, those are cell intrinsic, but they're able to align the phase of oscillation. So my lab studies basically two components of the system. The first component is how the clocks receive instructions from the environment and how those instructions can be manipulated externally to change cell behavior. And then the second component is the molecular mechanisms by which the clock components read these inputs and exerts their outputs. In other words, how they control cell physiology. And so those are the two themes that I'll be exploring today. And it's really exciting to be able to tell you a little bit about the applications of how what we learn can be applied for regenerative medicine. So speaking of regenerative medicine, we are in the business of making parts in the laboratory of the islets that control your blood glucose. We like to call them islet organoids for the following reason. We cultured stem cells from humans, so human embryonic stem cells or induced pluripotent stem cells, but we do so in a peculiar way. We cultured them in mass because we figured that for a human patient, you would not need thousands or millions, but rather billions of cells. So we have scalable protocols to make them in bioreactors. One of the ways that we do it is by in stir suspension bioreactors so that we can form cell clusters of about 1,000 cells each, where you can see here. And those clusters then receive instructions that we give to them by giving them signals to become, to differentiate explicitly towards endoderm and then explicitly towards pancreatic endoderm, and then to generate endocrine progenitors. And by now, instead of looking like clusters, they begin looking like mini pancreas. And in the border of the mini pancreas, we see a morphogenetic step in which the alpha and the beta cells that I've been telling you about are generated in order, the alpha first, the beta cells second, and therefore they form these little igloos with a mantle of alpha cells on the outside and a core of beta cells in the inside. Because the architecture of this resembles the endogenous architecture and the composition as well, we like to call those organoids, and that explains our term. So the cool thing about this is that you can treat diabetes by producing enough of them and transplanting them. This has been done actually in humans. There's our ongoing clinical trial for this. Typically for cadaveric transplantation, you do this in the hepatic vein. But one of the ways that this could be done in the future is by putting them under the skin in a less invasive way. And the objective of this is to go to somebody who depends on insulin injections for insulin control. This is typical in insulin-dependent diabetes, and to normalize their blood glucose through the transplants so that now they can live a life in which they don't need daily insulin injections. And they become a more normalized daily life. So this usually works with cadaveric islets, but there is a strong supply problem. And so if we could make them in the lab, we could solve the supply problem. However, unlike the cadaveric transplant, the stem cell transplant takes months to benefit the patient. And one of the reasons why it takes several months, unlike the primary tissue, is because the organoids are immature. So what we mean by them being immature, I give you a preview. This is a bit more of a technical definition of immaturity. you can flow glucose and increase from low to high glucose and then depolarize with a chemical, and then measure over time the amount of human insulin that organoids or primary cells give out in response to the increase in glucose. And then the reason why we say they're immature and under this definition is because the magnitude is very small compared to the primary mature cadaveric human islets. The kinetics are similar, but the magnitude is the same, and then the precision is also lower. And so we believe that there is a gap in the function of the tissue that's made in the lab, and if we could bridge this gap, we could make better transplants, but also better research models, better screening platforms, etc. The question, of course, then becomes, how do you bridge the gap? How do you go from immature to mature? And then one of the key things to observe is that they have the capacity to become mature because they can chemically be induced to act the same way. They just don't know how to do it physiologically or autonomously. So one of the things that we figure out to bridge this gap is that we could perhaps try to find what are the external cues from the environment that are necessary to become more mature. And we took a very computational approach in order to solve this problem. We figure out the cues come from outside on the environment. They invariably converge or are transduced into signal transduction pathways. Then they go to influence the function of protein factors in the nucleus that converge on the expression of target genes. So we can then figure out the target genes by RNA sequencing, and then we can work our way up by defining the DNA enhancer regions that control the stage and cell type specificity in the expression of those target genes, and then infer the regulators at those sites of control in DNA by combining chip sequencing and attack sequencing to infer chromatin and transcription factor regulators. And when we reconstruct circuits in this way, we might be able to infer what are the signal transduction pathways that are reading those cues. So this is an in silico approach. It's a little unbiased, but it might be familiar to people at the Broad. One of the things that this led us is to, for example, we compare circuits that we could reconstruct for beta cells before they are glucose responsive to the time when they are already glucose responsive, but not so mature. And so one of the things that we discovered or that we were interested in is motifs that are covered by nucleosomes in DNA that then become accessible to protein factors. So the proteins that enable these transitions to happen are considered pioneer transcription factors. And we're interested in discover who might be the pioneers that mediate the transition from not responsive to responsive. And to our surprise, we find an enrichment for the key core activators of the circadian clock, which are creatively called clock and ARNTL or BMA1. But we could also trace the expression of all of the components, repressors or activators of this feedback loop, and realize that most of them were not expressed in the progenitors and began to be expressed with the specification of the alpha or beta-like cells at much lower levels than the endogenous ones. So we said, these cells begin to have clocks with the specification of the endocrine cell fates, but they do not have them to the level that the endogenous cells have. And so we then began suspecting that the circadian clock factors can act as pioneers of the maturation process. Then one of the further clues that we could obtain in this direction is that when we reconstruct the circuit of the mature endogenous human cadaveric beta cell, there is a circuit that includes this DEC1 and DEC2 factors, which are circadian controllers as well, that were predicted to bind all of the previous controllers we knew were important for maturity, including PDX1, PAX6, NKX6, and MAF-A, which means that those players that through genetics we have learned are important to make a mature beta cells are actually heavily interconnected with the circadian clock, and not with the typical feedback loop factors, but with this more stage-specific factors, the DECs. So that also encourages us to ask the question, would it be possible to trigger maturation by triggering clocks? And then the question becomes, how do you get from A to B, from immature to more mature, if you can then activate the pathway? And how to activate the pathway becomes the next question. But this made sense because the immature cells, we know from studies of Lucifer's reporters of clock proteins, actually don't have rhythms when you're born, so 2 days postnatally in rats. But 30 days postnatally, they have robust rhythms that you can detect with oscillation of the proteins. And so it does seem that clocks are in the right place at the right time. And then the other transition that I had to talk to you about, when you go from more mature to less mature because of disease or genetics, this actually has been observed in rodents, in mouse and rat genetic models, but it's also observed in humans who are subject to shift work, in which case we know that disrupting the clock or disrupting the rhythms externally actually also reverts the maturity. So this made sense to us, but we still needed a way to trigger the rhythms. And so a clue came from another study in the lab, in the Melton lab, in which we actually profiled the serum of the mouse in the transition from the time where the beta cells are less mature to the times that become glucose responsive, to the time where they raise the threshold for glucose sensing, and ultimately, after weaning, we also profile the serum in the mice. And one of the things that we discovered by mass spectrometry is that the amino acid-derived metabolites were high before birth and then gradually decrease after birth, whereas the glucose-associated metabolite or fasting-associated metabolites, such as ketones, had the opposite pattern. They gradually increase after birth, and they especially increase after weaning. And so we've realized that this could be all a component of feeding and fasting. And in fact, this is published work. The amino acids have a higher composition in the environment before birth, and then the glucose takes over after you're born, but not in a stable way, rather in a fluctuating way, in which you have periods in which you have meals and periods in which you're fasting, explained in the rise in ketones. And therefore we thought, okay, so if this is what's happening in vivo, perhaps we can just mimic this in vitro in order to be able to manipulate the system externally. And so we did the following experiment. We took cells for 4 days and gave them 1 or 2 treatments. We fluctuated the environment periodically, or we changed the media, but we did not fluctuate between the basal and the fed state. And then we left the cells alone in media without feeding for 24 hours and measure their function by their insulin responsiveness. So as you can imagine, in cells that for 4 days you changed the media in a circadian manner, every 12 hours we changed the media, but the composition was stable, those cells had a stable response after the 24 hours in constant media. They had a stimulation index of the insulin in high versus low glucose that was constant over time. And those are our mock-treated controls. But when every 12 hours we switch between a feeding state and a basal glucose state for several days and then assess their function over a 24-hour period, we saw a dramatic time dependency in the insulin stimulation behavior. This time dependency is important because it is not different at the beginning and at the end of the oscillation, but it is severely different, tenfold difference in the middle of the circadian phase. So now you have 2 things to worry about. You have a time-dependent measurement that you cannot simply measure at any time. It has to, in order to observe the maximal difference, you have to be at the circadian peak. And then you also have the issue that you can do this through several inputs, not just with glucose, but with any other metabolic inputs, for example, insulin or amino acids or combinations therein. And so we could manipulate the rhythmicity of this behavior, not only in the organoids, which was our goal, but we could corroborate that cadaveric islets shipped to us on ice, which therefore had reverted in their maturity, could be made again to regain their maturity by this procedure. So this works in the stem cell derived, but it also works in the primary. But really the golden test, if we can control their insulin responses, is if we can make more mature tissues for therapy. And so the proof of fire experiment is to transplant them into a mouse and see if they're functioning. And the way we measure function is by human insulin in the mouse in response to a glucose injection. So in organoids that had not been treated with a fluctuating environment, the response after 3 days was minimal. You can detect human insulin in the blood, but it's not very responsive to a glucose injection. However, in tissues that had been trained with a fluctuating environment, you could actually see as early as 3 days a very strong response with a lot of variation, actually, mouse to mouse, that told us that this is a good avenue worth pursuing. And so it could make better transplants for function. in my own lab, this is published in my own lab, I became interested of what is the mechanism, okay? So if we can control the physiology of the cells externally, what does it depend on? What are the pathways that are mediating these changes at the molecular level? And so that's going to be the next part of my talk. I'm going to focus on what happens in the nucleus because, as I already told you, we were able to map networks of transcription factors. And we can actually rank them by their in degree, which is how many TFs are binding the regulatory sequence for a given TF, and by their out degree, which is how many genes of TFs they are binding to. So we could just add them in plus out, and the number one, the most interconnected TF from this definition of circuits is DEC1, one of the two circadian transcription factors that was densely interconnected with the maturity circuit. And so that made focus our efforts on that one circadian transcription factor because it seemed to be doing a lot of heavy lifting. We, by comparison, also studied DEC2. And so here's the two genes in more detail. They have several names. So you might have heard them through SHARP or BHLH or BHLHB factors. They contain a basic loop helix domain and an orange domain. There are only one gene in the fly, and it's called clockwork orange. And that circuit controls the clock by competing for binding to epoxes with the clock BMAL dimer. And as many clock genes, they're autoregulators, so they also control their own expression. So coming from the background of studying what happens in the nucleus, one of the first thing we wanted to do is to generate an in vivo phenotype to see if the molecular phenotypes translated into a physiology difference. And so we generated mice that are deleted for DEC1 in their beta cells, and those mice have elevated blood glucose, which means they're hyperglycemic. But if you give them a glucose injection, they also have a harder time clearing the glucose in a dynamic way from the blood, in a way that their heterozygote or just wild-type litter mates are not able to. This is a function of time. So the gene DEC1 is peaking during the fasting phase, which is the daytime for the mouse, and you see a large difference in their clearance rate. However, that difference disappears at night, but the gene is gone. And so it's also a time-dependent function. We're able to also corroborate that these mice have normal activity rhythms. That means they haven't lost their capacity to have a circadian rhythm to their life, to their behavior, to their eating, but they do have the expected impairment in phase resetting, so they do not reset the phase of the clock very easily. They have a deficiency in that. And then they also show a hypometabolic phenotype, which is lower energy expenditure. And so this is as opposed to the DEC2 mice, which had different phenotypes that I'll not speak about today. But suffice it to say that the islets of these mice actually form normally. The beta cells are made, the alpha cells are made, they're in the same arrangement with the same organization. They have the same organ size, the islet size. They have the same amount of islets in the pancreas, so the same islet mass. And so it's not a matter of the differentiation of the cells, but it is actually the time of development that we define as maturation, which is after the cells are made. We also check that the animals are not insulin resistant, so you can give them an insulin injection, and even though they start with a higher glucose because they're diabetic, they clear it at the same rate as a wild type. And so they, however, the one difference is that they're hyperinsulinemic, which means that if you take out their islets and give them a glucose injection, they have a much lower response kinetics. The kinetics are the same, but the magnitude is very different, just like the difference between the stem cell derived human islets and the primary human islets. These mice also have less mature islets. And so another measure of maturity is actually the kinetics themselves. If you expose to a gradual increase in glucose, this is called a glucose ramp. The mature cells are silent under basal and potently active under maximal stimulation. However, the immature ones actually respond to an increase at the basal level of glucose, but they have a poorer response in the maximal stimulatory condition. We can observe just this kinetics in these mice. And so the change in threshold and the change in absolute secretion is what we observe when we do this experiment in reality, glucose ramp. And we compare islets from the mutant mice, which have stronger response under basal levels, but blunted response under maximal stimulation levels. And so this let us know that precisely it's a problem with the behavior of the cells. They are behaving less maturely. but they are differentiating normally. And that's exactly what let us know that we're looking at physiologic maturation, and the window of physiologic maturation that we're looking at is here in the middle when the rhythms begin to be registered. And so this places the clock in the acquisition of the mature phenotype, which has to do with the beginning of glucose responsiveness at a high threshold for glucose in vivo. And so I think that this links the molecular background that we wanted. But we also wanted to come back to the nucleus and see what's going on. So we did chip sequencing, in which we can determine the binding sites for the DEC factors and compare them with the BMAL1 and clock. And as expected, we find that many of them bind the circadian rhythm genes as expected. However, they also have different targets. The DECs, the circadian rhythm is shared between the DECs and the core activators. But the control of insulin secretion and of energy metabolism in beta cells seems to be rather the job of the DECs rather than the core activators of the clock. So there's some specialization here, even though they're all bind boxes, which was very interesting to us, and we decided to investigate further. So the way we investigated this is a tough experiment for a graduate student. It means coming back every 4 hours to the lab for 24 hours, and then isolating RNA for sequencing, preparing the libraries, and comparing the rhythms of the genes. And so we found that 64% of the genes did not change in their rhythmicity by doing this experiment. 18% actually showed no rhythm, even though they were DEC1 targets. And then the quarter that did show changes was split between genes that no longer oscillate in the mutant. So to explain this a little bit better, each column is a measurement of RNA-seq, or the average of several technical replicates of RNA-seq at every 4 hours. And so there's an oscillation in wild type, in which different genes are peaking at different phases of the day, but that oscillation is lost in the DEC1 mutant. One of the curious things is that the oscillation is lost from the central activator of the clock, which is BMAL1, but not from the rest of the genes in the pathway. So again, there is a lot of specialization. However, the genes that belong, the functions of the genes that belong to this category were not exactly related to glucose sensing and insulin secretion. The ones that were genes that are not losing rhythmicity, but are shifting in phase. That includes genes that actually are responsible for the respiratory electron transport change, so TCA cycle genes, genes that are directly responsible for insulin secretion, exocytosis, and the fusion of the vesicles of insulin, and actually the mTOR pathway as well. And so genes that are responsible for both glucose sensing and insulin secretion were in this pathway. And we could plot the change in phase. If all the genes are in the same phase as the wild type, they would be clustered at 0. And each of these pathways, this is not GSEA, but PSEA, phase enrichment analysis. These guys are shifting 2 hours in the case of the TCA cycle genes, or 4 hours in the case of mTOR. And then some are actually shifting backwards. So instead of advanced phase, they have delayed phase, such as ATPase coupled calcium transport. And so if you think about this as gears, what we think is happening here is that the gears are not turning anymore in unison. They're not, the cogs are not clicking with each other, so that the capacity to mount a proper response to glucose sensing is impaired. And so that's our interpretation, but we also wanted to get deeper into what's happened at the single cell level in the nucleus. We did 3 types of omics, which you might be familiar with. One of them is single cell ATAC, another one is single cell RNA, and another one is called single cell proteomics by mass spectrometry. We are able to integrate these 3 data sets and hone down in the mature beta cells to see which is where we see the largest differences. And then we can intersect our analysis with the chip sequencing that we had before. And we did it in the following way. We asked for genes to be bound by DEC1, that is the green circle. We then ask for the genes to be differentially expressed. That means regarding of their oscillation, they will have a different mean expression level over time. And then we also ask them to be differentially rhythmic, so to have a rhythm that is altered. And when we do this, we can really now narrow down the playing field to about 31 genes, which are annotated to glucose responsiveness and insulin secretion. And the intersection of genes that are annotated for both functions is 4 genes that are known to be required and sufficient two of them are known to be required and sufficient for the regulation of the maturity phenotype. And so we know this because we can rescue the phenotype with chemical treatments. And so we believe that this is how we are seeing that DEC1 is responsible for synchronizing glucosensing and insulin secretion in order to achieve maturity. So the chemical studies that I was telling you about are a little bit complicated. The way that glucosensing and insulin secretion work is that glucose is imported through metabolism, ATP increases, this depolarizes the membrane, the calcium comes in, and then the calcium influx through pKa or pKc dependent pathways independently can lead to the fusion of insulin vesicles. So we can then tinker with the system by increasing calcium influx, and that actually does not rescue the phenotype. Then we can tinker with the system by putting a cyclic AMP analog for scolding, and that also fails to rescue completely. But one of the ways we can rescue the system is through PMA or carbachol, which stimulate the PKC pathway, and we believe that then we see no difference between the wild type and the mutant. So this is pointing to what step in the insulin responsiveness is failing, and we're very excited about following up with these results. But for the last part, I think, of my talk, let me summarize what we have and then tell you some new and published data. So we believe that feeding rhythms can be given to cells to manipulate the phase of their clocks, and DEC1 will be responsible for this phase manipulation in order to make the gears click at the right time of day so that energy metabolism can proceed and the cells can acquire a mature phenotype. And one of the consequences is we believe that if you apply this to other systems, you might be able to make better functioning neurons or better functioning cardiomyocytes in the lab. As I said, many of these is in preprints now. One of the things that we want to study next is how are the rhythms themselves generated. We understand how the molecular rhythms are generated, but we don't quite understand how the insulin responsiveness itself oscillates. And let me tell you what I mean by that. I come back to the map. This is a little bit of simplified map: glucosensing, metabolism, ATP, membrane depolarization, calcium influx, and then vesicle fusion. We have showed you that the insulin itself can be made to oscillate in vitro at the way it does in vivo, but we don't know which of these pathways mediates that function. And so we actually started to work our way backwards. So from the insulin secretion to the influx of calcium, we are able to measure calcium. And let's see if you can see this movie play. This is low glucose followed by high glucose, followed by return to low glucose, and then KCl to depolarize the calcium. And so KCl depolarizes the membrane that leads to the calcium influx. And we can simply measure the ratio of low to high at different times of day, low to high, which peaks at the 12-hour time point, which is precisely when you see the peak in insulin responsiveness. So that we know the insulin rhythm is preceded by a calcium rhythm. And so there's that step before the insulin exocytosis is also rhythmic. In order to measure one step removed, so the membrane depolarization itself, we need to measure electrophysiology. And for that, we have collaborated with the lab of GLU in Harvard. And then we have implanted tiny sensors in the developing organoids. And so the way we do this is we isolate the cells from the organoids, we plate them with stretchable mesh nanoelectronics, and then those electronics, since they're flexible, they actually become like a glove wrapped around the cells, and they become globally distributed. And what we can do with this type of tiny sensors implanted in organoids is that we can sense the activity of the electrophysiology activity of individual cells, and then expose them to high glucose, in which case they have higher activity. And by that metric, we can measure their function in real time at the single cell level in intact 3D organoids. And so we just had a bioarchive on that a week ago. And I think that one of the cool things that we can do with that is that in the beta cells or the alpha cells, we can measure the oscillation of the individual cells. And we can see that individual cells can be averaged, and you still see an oscillation, which means that the oscillation is happening in unison. They are all coordinated in the 24-hour oscillation. Over a period of 72 hours, we're seeing here oscillation in how much they are firing, how many action potentials they're giving per second. And then in response to high glucose versus low glucose for the beta cells, or vice versa for the alpha cells. But another thing we discovered is if you do a multidimensional analysis of the waveforms themselves are oscillating over time, and they have the night phase and day phase. So it's not just the behavior of the cells, so it's not just the system level, but it's also the cell level. The action potential wave itself is oscillating. And so we now know that these 3 steps are in unison in order to create insulin rhythm. But actually, if we go back to the beginning of what triggers insulin secretion, which is metabolism and ATP, surprise, surprise, we also find oscillations there. We can measure in 2 ways. We can measure respiration. So here in the first plot, I have deconvolved the insulin secretion in response to high glucose, in response to KCL, or on their base of glucose. And then here in the center, I have plotted the dissolved oxygen in the media, which peaks around 12 hours, precisely at the point of peak insulin secretion. We can also measure the CO2 in the media using a blood gas analyzer, and then it actually dips at the circadian peak point. So we know these cells are respiring in unison as well. But we can go deeper and actually image the mitochondria inside of single cells. And we've done this around the clock when we observe 2 things you can observe in this Z projection. First, that the mitochondria begin as circles and end as circles, but in the middle, they become something rather different, which is a branched network of mitochondria. And the period, the time, the peak of mitochondrial branching is precisely at the 12 hours, which is a circadian peak or insulin function. So in cartoon form, what I'm depicting here is when the mitochondria are more branched, which you can see here at the circadian peak point, they consume more oxygen, which you can see by the dissolved oxygen in the media. They also produce more reactive oxygen species and ATP, which we're able to measure. However, when they are in the more fragmented network form, so they look more like circles at the end of the circadian cycle, they are actually less active. They produce less oxygen, less ATP, and less reactive oxygen species. And the point is that there is an oscillation between the 2 states that happens in a circadian timeline. And this oscillation is controlled by a protein called DRP1. This oscillation, this protein itself oscillates. In the wild type, it peaks at the 12-hour time point. And in the mutant, in the DEC1 mutant, that oscillation is lost. So we believe that part of the mechanism, how the GSIS, the insulin responsiveness, is lost is through the loss of the mitochondrial oscillations that then give you electrical oscillations, calcium oscillations, and insulin oscillations. And so we believe that that's more or less the entire pathway. All right, so in my last slide, I want to leave you with some thoughts for the future. Many of you have very different projects, but all of you can ask yourselves, should we give cells meals? Should you be feeding your cells not constant composition of nutrients, but a fluctuating composition of nutrients in a given pattern? That's food for thought there. Another food for thought is, when should you measure the phenotype that you're measuring? Whether you measure cells isolated in an incubator or mice, should you measure in the morning? Should you measure in the night? Should there be a difference? And how do you know if there is no difference? And then the third question is a translational one. You know, when we give drugs, should we give them at a certain time of day when their targets peak, for example? Or should we drug the clocks themselves so that we can apply drugs at any time we want and have the same efficacy? And so I think that those are interesting things when we do screens, for example, and we do them in vitro, and we conclude that something is working or something is not working. Is that a time-dependent function itself? So I think these are hard questions, but they're interesting to study. And then I'll stop there. I'll acknowledge the people that have contributed to this many studies, beginning with my postdoc lab, then this lab of lab, we did the mass spectrometrist, the Liu lab, which we contribute with for single-cell electrophysiology, the people in my lab currently, and people at Penn that have helped with this work, as well as the institutes and foundations that support our work. Thank you.
That was amazing. That's really impressive. And I like how you integrate functional annotations and different measurements for electrophysiology, and you're mixing in, you know, organoids and functional genetics and circadian rhythms and single cell. It's really impressive. I just want to mention that we have a short survey, so I'm going to put that in the chat. If you can fill out the survey, that'd be great. And I'm going to open the floor for questions. And just a quick question from my end. So single cell is, you know, it can be very noisy, and circadian data can be very noisy. how are you dealing with the noise in your data? And have you thought, well, a second part is, have you thought about using methylation as a marker? And how is it going to change in an environment like a petri dish as opposed to a cell?
Right. So those are two different questions. And so I think that to answer the first one, we measure oscillation with confidence in bulk. We don't measure oscillation with confidence in single cell. So when I show you RNA sequencing oscillation, that comes from bulk RNA sequencing, actually, because then we have a very nice dynamic range, and we can sequence all of them at the same time, right? There's some immunization batch effect, and we can use tools that have been created to infer rhythmicity in bulk data sets. Our actual single cell data sets, so in terms of sequencing, they're snapshots. They're not continuous every 4 hours for a number of reasons, including the cell encapsulation that you would need to do that every 4 hours. But we're working towards. But there's actually a lack of tools to infer rhythmicity from single cell data. You can post hoc infer rhythmicity of if you know when patient data was collected at the single cell level, but I don't think there's a lot of rhythm inference that can be done. Rhythm inference is very complicated. It involves model feeding, or it can be a Bayesian endeavor, and I don't think there's generally accepted ways to do this with single cell data sets. Now, in terms of the electrical single cell measurements, I think it becomes rather easy because the cells are synchronized, and therefore we can, if we see a rhythm in bulk, we don't need to actually go see the rhythm at the single cell level, but we can through the fate analysis that we did. Now, your second question, which is methylation, we have no idea how dynamic methylation is over 24 hours. We know it's dynamic during development. We know it's dynamic with gene activation and gene repression. So perhaps there will be a subset of methylation events that will be circadian. Or perhaps that is already known. I actually don't know.
Well, yeah, from my experience, it's been very noisy with circadian, even in bulk. So
from your experience has been what?
Even in bulk RNA-seq, it's
right. It can be incredibly noisy. I can imagine. We have a project that looks at chromatin oscillating over a 24-hour period, and it's challenging.
We have a question in the Q&A. From Carmen Lee, amazing talk, Juan. Do you think the circadian rhythm dependency of other cell types beyond the pancreatic islet cells will also be metabolic in nature? For example, in neurons?
Yeah, there is this intrinsic link between the clock and metabolism. And that link might be evolutionary. It might be that before cells had nuclei, the oscillations came from metabolism itself, at some point from mitochondria. And so the clock proteins themselves bind metabolic genes and control mitochondrial cycles. There's feedback in both directions. So I do expect that all the cell types in the body will have circadian functions to clock protein and non-circadian metabolic functions for clock proteins. So I think there's quite a bit of research in neurons about calcium handling and metabolism for the circadian proteins. I think the real important question is, is the maturation going to be also controlled through the clocks in the neurons in the way that it is in metabolic tissues, or at least in this metabolic tissue? But one of the caveats that I always have to mention is that when we give a stimulus every 24 hours, if that stimulus is metabolic, there is a chance that the effectors, the downstream effectors, are not circadian proteins, but are just metabolic proteins. And in fact, it's quite likely that we should call our daily feeding entrainment that instead of a circadian entrainment, because we don't strictly know that this is dependent on circadian proteins themselves.
Another quick question for you. Have you looked at RNA velocity and what kind of genes are being up and down regulated, like how the transition points are going for some of these metabolic processes, especially in the context of circadian rhythms?
Yeah, let me see if I understand correctly. So RNA velocity at the single cell level, or in the
single cell, or the pseudobulk level even?
At the pseudobulk level. I think that would be interesting. I don't think we've done that, but I think that one of the things that could come out of it is to see, well, again, the data is very sparse, right? In most single cells. So I don't know how much we would get out of it, but I think it's interesting looking into that. And then you were saying, what was the second part of it?
Oh yeah, like because you have the RNA velocity, and you can tell sometimes what genes are being up and down regulated based on the almost like a vector, right? A directionality vector. But because you have such oscillation in the circadian rhythm, and depending on the cell type, you might have more frequent oscillations than day and night. It might be hourly or timed at meals or something like that. It might be interesting to take a look at.
Yeah, I mean, this is a very complex subject, but let me say, I'm sorry to gloss over that, but let me say that about 80% of the transcriptome oscillates with a 24-hour period, depending on where you look, right? So if you look in a given cell type, you'll see 10 or 20. But if you then look at the rhythmic genes across, let's say, 200 tissues, then you almost saturate the transcriptome. Almost every mRNA oscillates. It is true that there are many clocks. So there's a 24-hour clock, and that's important for the sun rotation around the planet. But there's also a 12-hour clock, which is important for the moon rotation. It's called the Circadian clock. There's 4-hour clocks like the segmentation clock during development. And there's many clocks. There's also ultradian clocks. So animals that follow hibernation or migration are able to keep time of the seasons of the year autonomously without external input. So yeah, I think that there'll be quite a lot of things to discover there. So pretty exciting. So if we don't have any more questions, I'll leave a couple of minutes. Yeah, we can adjourn the meeting. We're up against the hour right now, but thank you once again for an amazing talk. Very interesting stuff. Thank you, Ricardo. Thank you all for coming.